waktu apaan ini ya? Diandra jadi juga apa tuh? Semua ini. Nanti semua gabung tuh nggak apa-apa tuh. Jadi takut internetnya down gitu loh Bapak. Ya nggak sih?
Ya kan ini gelar dulu.
Hilal, salsa.
Ini gelar. Aku cukup tadi. Makanya harus di oke dulu.
Siapa? Terus habis hilal, salsa. Ya udah mending sekalian semua, kalau join tapi nggak jadi apa-apa kan kayak sayang ya. Ya. Apa? Kak Norman. Terus Anggun. Afki udah gabung? Oke. Kok nggak bisa ya, Afki? Udah. Apa? Oh.
Universitas Negeri Malang offers the best education, research and community service in Indonesia. UM has consistently been maintaining its position as one of the best universities in the country. In 2018, UM is in the top 15 universities in Indonesia according to the Ministry of Research and Higher Education and 4ICU, which proves its quality as a center of excellence and reference. UM has dedicated itself as the learning university since 2009. It has strengthened its vision as a learning organization and as an outstanding learning resource in Indonesia. UM students are distributed to various high-quality and well-reputed study programs, extending from associate degree to doctoral degree programs. Among UM students, there are international students who come from various countries across the globe. Recently, UM has 45 study programs accredited B and 59 study programs accredited A nationally. UM was established on 18 October 1954 as a College of Teacher Training and Education, PTPG. The institution transformed into UM on the 4th of August 1999. UM's main campus is in Malang, surrounded by education facilities, recreation and tourism spots, sports and business centers. UM's Campus 2 is located not far from the east gate of Malang. UM's Campus 3 is located in the Gate of Heritage area of the Shrine of Sukarno, the first President of Indonesia, in Blitar. Faculty of Education, Faculty of Letters, Faculty of Mathematics and Natural Sciences, Faculty of Economics, Faculty of Engineering, Faculty of Sports Sciences, Faculty of Social Sciences, Faculty of Psychology Education, Postgraduate Programs. UM also offers associate degree in vocational education. They provide trainings in various skills. These are managed under the skill training programs for automotive, business, industry, and information technology, or known as PROBIS. Under the guidance of lecturers and experts in their respective fields, students can experience revolutionary learning innovations, develop their critical thinking skills, produce innovations, and nurture their enthusiasm for knowledge. These endeavors perfectly fit with UM's aspiration in becoming the best center for learning innovations in Indonesia. Universitas Negeri Malang is always committed to making a better Indonesia. At UM, we learn, do research, serve the community, and contribute to the betterment of Indonesia. UM, the Learning University. Universitas Negeri Malang offers the best education, research, and community service in Indonesia. UM has consistently been maintaining its position as one of the best universities in the country. In 2018, UM is in the top 15 universities in Indonesia according to the Ministry of Research and Higher Education and 4ICU, which proves its quality as a center of excellence and reference. UM has dedicated itself as the learning university since 2009. It has strengthened its vision as a learning organization and as an outstanding learning resource in Indonesia. UM students are distributed to various high-quality and well-reputed study programs, extending from associate degree to doctoral degree programs. Among UM students, there are international students who come from various countries across the globe. Recently, UM has 45 study programs accredited B and 59 study programs accredited A nationally. UM was established on 18 October 1954 as a College of Teacher Training and Education, PTPG. The institution transformed into UM on the 4th of August 1999. UM's main campus is in Malang, surrounded by education facilities, recreation and tourism spots, sports and business centers. UM's Campus 2 is located not far from the east gate of Malang. UM's Campus 3 is located in the Gate of Heritage area of the Shrine of Sukarno, the first President of Indonesia, in Blitar. Faculty of Education, Faculty of Letters, Faculty of Mathematics and Natural Sciences, Faculty of Economics, Faculty of Engineering, Faculty of Sports Sciences, Faculty of Social Sciences, Faculty of Psychology Education, Postgraduate Programs. UM also offers associate degree in vocational education. They provide trainings in various skills. These are managed under the skill training programs for automotive, business, industry, and information technology, or known as PROBIS. Under the guidance of lecturers and experts in their respective fields, students can experience revolutionary learning innovations, develop their critical thinking skills, produce innovations, and nurture their enthusiasm for knowledge. These endeavors perfectly fit with UM's aspiration in becoming the best center for learning innovations in Indonesia. Universitas Negeri Malang is always committed to making a better Indonesia. At UM, we learn, do research, serve the community, and contribute to the betterment of Indonesia.
UM, the Learning University.
Excellencies, the Dean of Faculty of Mathematics and Natural Sciences, Universitas Negeri Malang, Professor Dr. Hadi Suwono, Master of Science, distinguished guests, Professor Ernest Frankel, PhD, Mario Guaracino, PhD, Riza Putranto, PhD, participants, ladies and gentlemen, good morning, and Assalamualaikum warahmatullahi wabarakatuh. We are delighted to welcome you all in this Zoom meeting platform to attend this event of International Bioinformatics Lecture held by Department of Biotechnology, Faculty of Mathematics and Natural Sciences, Universitas Negeri Malang. I, Norman Yosi Haryono, am the Master of Ceremony for today's event. First and foremost, I would like to ask myself and all of you to lower our heads for a moment to say a little prayer as a token of gratitude toward the Almighty. Let us pray together. We may begin. Enough? Now we are going to have to listen Indonesia's national anthem, Indonesia Raya. For the operator, please assist me to play our national anthem.
Indonesia
Excellencies, distinguished guests, participants, ladies and gentlemen. Now we are going to listen to the opening speech delivered by the head of the committee, Kenis Rozana, Master of Science. To Ms. Rozana, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, first of all, I would like to thank you for the many parties involved in today's International Bioinformatics lecture. For the amazing speakers, all participants, including the online course Bioinformatics participants who also took part in this event, and not forgetting for all committers who have worked hard. International Bioinformatics lecture today was attended by more than 500 participants and come from almost 7 countries, namely Indonesia, Malaysia, Singapore, Afghanistan, Kazakhstan, and the USA. But now I see the participant may be below because I think it is some countries have different time, so I think it is the difficulties in this event. Okay. Actually, I'm not focusing on the number of our participants, but I think the number of participants reflects the usefulness and attractiveness of our events. I know organizing this event is not easy. Asking for the speakers opportunities who are in different places and times is also challenging for us. But all of that paid off with the appreciation of the ladies and gentlemen who attended this event. As I know, bioinformatics in research is like the internet in our life. Maybe we can live without it, but will be better with it. As the chairman of the committee, I hope the International Bioinformatics lecture today can be useful for all participants. If you have any comments or suggestions regarding our event, you can contact us anytime. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Rozana, for the opening speech. The next opening speech comes from the Dean of Faculty of Mathematics and Natural Sciences, Universitas Negeri Malang, Professor Dr. Hadi Suwono, Master of Science, who already here with us. How are you, Prof. Hadi? I hope you're in the good condition.
Yes, thank you, Pak Norman.
Thank you, Prof. Hadi, for joining us today.
Thank you.
Without further ado, to Professor Dr. Hadi Suwono, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable keynote speakers, first, Professor Ernest Frankel, PhD from MIT, USA. Good evening, Prof. Ernest. Second, Mario Guarasino, PhD from University of Cassino, Italy. Good morning, Prof. Mario. And Risa Putranto, PhD Director of Indonesian Society of Bioinformatics and Biodiversity. Good morning, Dr. Riza, Honorable Professor, Lecturer, distinguished guests, and all participants. As the Dean, Faculty of Mathematics and Natural Sciences, I appreciate the participation of all parties in holding the International Bioinformatics Lecture today. As a newly established study program, I hope that the Biotechnology Study Program can always organize events that attract everyone's interest, both at the national and international level. As a part of higher education, biotechnology under the auspices of the Universitas Negeri Malang is always determined to provide the best service for the entire academic community and the wider community. Hopefully, this International Bioinformatics Lecture can be a driving force for other more significant events to provide a space for communication and sharing of knowledge that continues to develop. Therefore, I would like to give my sincere appreciation and gratitude to notable keynote speaker. Please let me extend my deepest gratitude and highest appreciation to all committee members. Participants from 9 countries for attending this bioinformatics lecture. I hope that this bioinformatics lecture will provide useful knowledge, insight, and experience, as well as improve the quality of research. Finally, in the name of Allah Subhanahu wa Ta'ala, and all praise belong to Him for granting us the opportunity and academic empowering. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Professor Dr. Hadi Suwono. Excellencies, distinguished guests, participants, ladies and gentlemen, we are now moving into the main session of this event, which is the presentation from our invited guests who are kind enough to share their expertise and knowledge to us all. But before that, I would like to introduce the moderator for the main session, Dwi Listiorini, Master of Science, Doctor of Science. Ms. Listiorini earned her bachelor degree in IKIP Malang, and she earned her master degrees in Bandung Institute of Technology, and she earned her Doctor of Philosophy in Tokyo Metropolitan University. Some research that she's been doing in these years, including taxonomy study of hourglass frog, Leptophryne bourbonica, in East Java region with mitochondria 16S rRNA gene and morphological characters. And the second is single nucleotide polyformism analysis of COL1A1 and FGFR2 genes and visualization of protein conformational changes as marker of skeletal maloclusion phenotype. Some publication that has been published in this year, including the first one is the comparison of SARS-CoV-2, SARS-CoV, and MERS-CoV genome and spike protein variation, and the second one is identification of potential bacteria on several lakes in East Java, Indonesia, based on 60 sRNA sequence analysis. Excellencies, distinguished guests, participants, ladies and gentlemen, without further ado, please welcome Ms. Listiorini.
Thank you, moderator. Dear participants and all, it is great to see you, to meet you in this morning, in this occasion. Ms. Listiorini already been introduced by Mr. Yossi, and here I will be with you to moderate as a moderator for the presentation from three speakers: Professor Ernest Frankel from MIT, USA, and it will be continued with Professor Mario Guarancino from Cassino, Italy, and Dr. Risa Putranto from MABPI, Indonesia. We will have this presentation in the panels, and the discussion will be conducted after the presentation of three speakers we have this morning. Before we start with Professor Ernest Frankel presentation, let me introduce Professor Ernest. He is the professor of biological engineering from MIT, USA. He developed the Frankel Lab to conduct their researches, and their research mostly in the, system biology of disease, computational biology, neurodegenerative disease, diabetes, and cancer. So his work and he and his colleagues brought from the metabolic disorder up to neural sciences problem. And Professor Frankel received his bachelor in from MIT and also his PhD. He already published a lot of papers, of course, and we compiled at least 33 great ones, and 2 of them are in genome scale screen identify factors regulating the tumor cell responses to natural killer cells and immortalized striatal precursor neurons from Huntington's disease patient derived iPS cells as a platform for target identification and screening for experimental therapeutics. This time, he will present for us his work, and it will be spent for 30 minutes. So let us join the presentation. Operator, please. It seems that we have a little technical problem. We can wait a little.
Diseases of the brain. There's been a long history of technology making amazing progress in treating disease. And what I show here is a graph of deaths in the United States. I'd like to thank the organizers for inviting me to speak in this lecture series and to give this International Bioinformatics lecture. I apologize that I'm not able to join you in person. My topic today is using new technologies to understand disease, and in particular, in whether artificial intelligence can help us understand diseases of the brain. There's been a long history of technology making amazing progress in treating disease. And what I show here is a graph of deaths in the United States per 100,000 people over the course of the last century, the 20th century. And you can see a number of remarkable trends. You can see a pretty strong decline in the early part of the 20th century. When we break this apart into both infectious and non-infectious diseases, you see something even more amazing. At the beginning of the 20th century, about half of all the people who died in the United States died of infectious disease. And by the end of the 20th century, those numbers were a tiny, tiny fraction of all the deaths. Now, progress is not always uniform. We saw a tremendous setback here in 1918 with the Spanish flu, but the technologies that allowed us to conquer these diseases were really quite diverse. And some of them are things we don't even typically think about as issues of biomedicine. So some of the things that were really fundamental in the early part of the 20th century were sanitary engineering and food science. And the antibiotics that came in mid-century were really a tiny piece of the puzzle, but a very important one. I mentioned already, there was obviously a spike of deaths in 1918 with the Spanish flu, and we're seeing again a spike of deaths, excess mortality due to COVID-19 in the 21st century. But the core technologies we need to conquer COVID-19 already exist. And the question is, what can we do about all the diseases for which we don't currently have technologies? Are there ways to bend that blue curve, perhaps, as much as we bent the red curve? I'm going to focus today on dementia. This is a worldwide problem. These data from the World Health Organization show that is one of dementia, Alzheimer's in particular, but other dementias as well, are one of the leading causes of death throughout the world. And in the richer countries in the world, where are the resources to deal with some of the more preventable forms of death, we see something quite interesting, that while we're making progress in rich countries with the resources in things with like heart disease and stroke, we actually have a growing problem of Alzheimer's and other dementias. And so this problem will eventually come to all of the world as we get the resources in the rest of the world to deal with other currently preventable forms of death and disease. So the question I'd like to pose today is, how can technologies help us with dementia? And the key here is to really try to understand what's worked in the past. What lessons can we take from the diseases that we do have the technology to conquer? In particular, we can learn a lot from cancer. One of the things that's been really critical is obviously understanding the cause of disease. That's been true of COVID. Our ability to rapidly identify the virus obviously has allowed us to develop the vaccines that are now making their way around the world and will contribute tremendously to reducing the death and suffering from that disease. So we need to know the cause of the disease. And I've drawn it here as a bullseye, a target that we're going to try to aim at with molecules to try to prevent the spread of the disease. And then critically, we have to test a lot of ideas, a lot of shots at this target, because not everything that we try is necessarily going to work. We were very fortunate in the case of COVID that some of the early ideas that were tested in terms of vaccines did work, but we'll see that in other diseases, the progress is definitely going to be a lot slower. Now, if you look at another infectious disease that we've been dealing with now for decades, HIV/AIDS, it's a virus that infects cells, and it goes through a viral life cycle that's shown here, where it first enters the cell, it inserts its genetic material into the cell, which interacts with the genome that's already present. And then it turns the cell into a virus factory that produces more and more virus. And what worked to prevent the spread of HIV was developing drugs that focused on key elements of this viral life cycle. So there are a bunch of molecules that I've outlined here that attack key molecular transitions, parts of the viral life cycle that are necessary. So by identifying each of these targets, society was able to make drugs that hit those targets that block the virus from progressing. This is a recipe that clearly worked in the case of HIV, But in some ways, infectious diseases, despite the tremendous devastation that they can cause, are the easy case because there's something from outside the body, something very different from us. And so the enzymes, the targets in the virus, can be distinguished more easily from those in the host, in our own bodies, than for diseases that arise without an infection. But can the same kind of thing work for cancer? Well, we do know that it can. A number of years ago, there was really a remarkable development in a certain kind of cancer, which we know is caused by a particular combination of two genes that come together. These are genes that are normally present on different chromosomes, chromosome 9 and chromosome 22. One of these genes we call BCR, the other one we call ABL. And in some people, there's a transformation of the DNA that brings these two genes, which should be completely separate from each other in the genome, together. And in patients that have this fusion of these two genes called now BCR-ABL, they develop a disease called CML. And CML is a pretty devastating disease. But once it was understood that it's caused by a unique gene that's not present in the rest of the population, we're able to develop drugs that treat this disease. And so the question is, can we do the same for dementia? Can we find those targets like the one in HIV, like the ones in CML, that allow us to distinguish what is causing the dementia and then develop drugs to target it? So one approach to do this is to look at a lot of people, some of whom have the disease and some of whom don't, and look at whether there are any genes that are different in the people with the disease from those without the disease. That's what got us the results, for example, in the CML cancer that I just talked about. And so we can try to find out whether there's something unique in the genomes of the patients. And what we find then would be a genetic cause. Now, it turns out that in dementia, there are some rare cases where a single gene does cause the dementia. And in Alzheimer's disease, it's estimated that about a quarter of the people who develop the disease do have a variation in their genome that is the cause of the disease. But it turns out that the vast majority of people with Alzheimer's, and this is even more the case with some other diseases, don't have a unique gene that serves as a good target for the disease. They have what we call risk alleles. A risk allele is something that somebody may have that may increase their chances of getting the disease, but there are many people with that risk allele who don't get the disease, and many people with the disease who don't have the risk allele. And so it's been very, very hard to figure out what targets to use in diseases like dementia, Alzheimer's, and others, where so much of the disease doesn't have a unique genetic origin. Well, we can learn one more thing from cancer. It's not just about finding a target, but it's about figuring out what disease we're dealing with. A long time ago, people thought of cancer as a single disease that could be treated with fairly uniform treatments. This old poster from the 1930s said that there were only 3 ways to treat cancer, and it treated all cancer the same: surgery, X-rays, and radium, kind of radiation. Now, we understand these days that we can't treat all cancers the same. And in fact, when we think about cancers, we often divide them into the tissue that's diseased. So maybe it's a lung cancer or a skin cancer. But that's not enough. We know that even within those subtypes of disease based on tissue, there are subtypes based on molecules. And so people who have studied breast cancer have been able to divide breast cancer into a number of different subtypes, each of which needs a different kind of treatment. This is the key insight, being able to not only know that there are diseases that have targets, but figuring out that those diseases sometimes look to the doctor like they're one thing, but when we get the right molecular tools, we discover there are actually multiple different diseases. And once we're able to break apart cancers into different kinds of breast cancer and different kinds of lung cancer, then we can find the right targets for each subtype of the disease. So compared to dementia, actually cancer is actually relatively easy because the subtypes are easy to find. Typically tumors get cut out of the body. And so we can then take those tumors into the lab and study the molecules in very, very great detail to find out what the subtypes are. But we can't easily subtype patients with dementia or other neurodegenerative diseases by taking out their tissue and looking at it in the lab. That's much too dangerous. Many of these diseases, if you went in and biopsied them, you cause more damage than help. Similarly, when we think about the targets that we need to go after in cancer versus dementia, they're very different kinds of things, because in cancer, the targets that we're trying to go after are things that may be a unique thing that turns a normal cell into a cancer cell. And so these are really, really clear hallmarks of disease. But in neurodegenerative diseases, it typically isn't so clear-cut. The things that most neurodegenerative diseases occur over long periods of time. And so it's a slow failing of the biological system. There may not be one unique cause that leads to these dementias, and may be an accumulation of multiple problems. And finally, when we think about how to hit the target, well, in cancers, what we're trying to do is kill a dangerous cell. The same thing is true with viruses. We're trying to kill off the virus. But in neurodegenerative diseases like dementia, we're trying to keep a sick neuron alive. And that is probably fundamentally a much harder task. So we know that we've got our work cut out for us, but still we can take a lot of insights from what we've learned from these other diseases, infectious disease and cancer. So to summarize what we've talked about so far, new technologies have shown that we can dramatically change the course of human health. We can eliminate certain diseases completely and bring other ones to bear so that they become manageable diseases. We need the right technology for each disease. A part of this is going to be finding the subtypes of the disease. And then once we've found the subtypes, figuring out what the targets are. So from this, we can come up with a roadmap to cure neurodegenerative diseases. The first thing we need to do is gather a lot of data. This will help us understand the subtypes of the disease. We've got to take those data, develop new technologies to make sense of them. Artificial intelligence is going to play a critical role here. And then once we have the subtypes identified, then we can start to identify the targets and figure out what are the right drugs. And we can use different tools from AI, things called causal modeling, to help us engineer new treatments. This is a vision that we have, and we've started to apply this vision to ALS. Amyotrophic lateral sclerosis is a disease that some of you may have heard of in context of certain famous people who've come down with it. Here are 3 different people who are known around the world. Lou Gehrig was a United States baseball player. Stephen Hawking is a famous physicist in the United Kingdom, and Mao Zedong in China. All of these people suffered from ALS, but the course of their disease, each was unique. What happens in ALS is that the neurons that transmit information from the brain to the muscles slowly fail. It's a progressive disease, which means it comes on slowly. The initial symptoms are often mild, and it can often take quite a long time to get a clear diagnosis as to what the disease is. But invariably, it's fatal, because ultimately, the body loses control over breathing. The neurons that control the lungs, the diaphragm, eventually fail. So we've taken upon ourselves to try to figure out how we can advance the search for a cure for ALS. And as I said, the first step in the process is gathering data. And so we've tried to gather all the data we can from around the world. We cooperate with many, many different organizations that have collected data. Some of it, like Project Mine and GTAC, is genomic data. Others, I'll tell you quite a lot about Answer ALS, and perhaps a little bit about Everything ALS, ALS TDI, organizations that try to collect many different kinds of data beyond the genomics, information about molecules, but also information about clinical patterns in patients and behavioral patterns. And so we've gathered all that data. In the Answer ALS program, we've decided that we need some way to really understand the neurons from these patients. But as I've already mentioned, we can't biopsy it. We can't cut out neurons from a healthy patient. And once the patient has unfortunately passed away, the tissue that we're looking at represents late stage of the disease. It doesn't reflect the stage of the disease where drugs are likely to be able to help. So we need some way of actually studying patients' neurons before they pass away. And here there's an amazing new technology that allows us to tackle this. We take a blood sample from a patient or from a healthy person, and in the laboratory, we can transform it into something called an induced pluripotent stem cell. This means that we take that blood cell that used to be just a blood cell and make it into something that could give rise to lots and lots of different kinds of cells. And then we can coax it in the laboratory into becoming a neuron. And in principle, we can do it into many other kinds of cells as well. And now we've got a limitless supply of neurons grown from cells of a particular person. And we can study the consequences of the genes in that person and other factors in a neuron that's from a living neuron of a human being, even though it's not directly taken from the person as a neuron. Another kind of data that we can gather are information about medical records and also behavior. So electronic medical records are becoming more and more common throughout the world. It's possible then to get very detailed information about the progress of the patient's disease. But that's only when they're interacting with their physician that we get the electronic medical record. So we'd like to get information about what happens to people when they're not in the doctor's office. Perhaps it could be months or maybe even years in some diseases between when somebody visits a doctor. We need to get information about that. And now we can get a lot of that information by using cell phones or other tools to interact with people over the internet. And we can ask these patients and volunteer controls to participate in studies and give us information about how things are progressing in their lives, be it fine motor skills, like how well they're able to trace figures or write, to gross motor skills, how much they're moving around, or their speech. All of these things can give us deep insights into the disease. One thing we'd like to then be able to figure out are the subtypes of the disease. And maybe the obvious subtype you can think about is how fast someone progresses through the disease. There are some people who pass away very quickly after developing their first symptoms, and other people who take quite a long time and survive the disease for quite a while with only minor changes. And you can imagine mapping some clinical measure of how serious the disease is on the vertical scale, and then time on the horizontal scale. And we'd like to be able to separate patients who have this kind of slow response seen for the green patients from this more rapid response seen by the patients with the beige or orange symbols. But actually, it turns out to be rather difficult to separate slow and fast progression, because a lot of the ways that we currently measure the disease are not that precise. And also, we don't get to talk to the patients, see them for long periods of time. And so what we end up seeing is rather short windows into the life of the patient, just a little bit, maybe 2, 3, 4 visits. And during that period, we're asking them questions to assess the severity of their disease, how they're feeling, how well they slept, how well they were able to dress themselves or cut up their food. And what we find is that when we ask these patients questions, the results that we're going to get are depending on all sorts of things. Some things, they'll have bad days or bad weeks, but then those bad days or bad weeks will go away. And so we may get noise in our measurements that make it very hard to tell that the green patient was a slow progressor and the other patient was a fast progressor. And so we've been developing artificial intelligence-based methods that allow us to take these noisy data and identify groups of patients who actually have fairly similar trajectories across. And so on the right-hand side are real data from one of these studies, the ANSWER ALS study, that allowed us to identify, despite all the noise, patients with slow progression and fast progression. And so this is our first insight into the patterns of the disease. And what we found is that patients actually, from different studies conducted at different times in different parts of the world, actually show similar patterns of decline. Some patients show very rapid decline with then a tail where things start to stabilize. Other patients show very slow linear progression. The difference between the slow linear progression and these other have a huge implication for how we think about clinical information and clinical trials. So you can see in a study like this that even subtle shifts from the initial linear progression, we would have imagined these patients would all decline fairly rapidly, but in fact, all of them took a turn for the better and progressed very slowly. Now, in patients who have suffered from all these different possible combinations of clinical parameters, and we'll go through these diseases differently, go into the same clinical trial, it turns out it's very hard to tell when a drug is working or not. I'll show you data here from a clinical trial that failed. And what we see here are the patients going through the trial, and just a subset of them that show the most nonlinear behavior. And unfortunately, a lot of the modeling that's done to decide whether a drug progresses is based on a linear prediction. We draw a straight line and say, we expect that the patients will continue to progress along this straight line. But in fact, that's not the case. Quite often, there'll be dramatic deviations from linear. Sometimes it leads these clinical trials to underestimate decline, and sometimes it leads us to overestimate decline. And the more accurate kind of modeling that our lab has produced now will allow us to then more accurately go back into clinical trials and hopefully find patients who are naturally going to be more like the red ones, and other patients are going to be more like the blue ones. and treat them differently in our statistical analysis to be able to tell whether drugs are actually working. That's part of the problem, is the clinical, but we also need to do a deep understanding of the molecular changes. And the Answer List program has gathered 1,000 patients from whom we not only have the clinical data, but we also have, as I told you, the behavioral data. And then using these iPSCs, these neurons derived from blood of the patients, we've gathered a lot of information about their proteins, their metabolites, well, actually not metabolites, I should have said here, but their transcriptional data, their epigenomes, and their genomes. And we then use artificial intelligence methods to gather all that data together and identify subtypes of ALS. So far, we've focused on one known genetic type of this disease, people who have a mutation in a gene called a C9orf72, or C9 for short. And we've taken neurons from those patients, we've gathered their genome, their epigenome, and the proteome. And then we've used these AI-based methods to figure out why does a variation in the C9orf72 gene cause disease. And we're able to identify lots and lots of molecular consequences of this genetic variation. Some of the things that change involve changes in how molecules go between the nucleus and the cytoplasm of the cell, how proteins and RNAs interact with each other. These kinds of changes on the left-hand side we've been able to show are things that cause the disease, cause the patient's cells to work worse, and eventually cause them to lose function. Interestingly, there are other changes that are occurring in the cell that actually are good for the person. One of those things is the stress response. So when you get the defects on the left-hand side, the body naturally tries to compensate. And the changes on the right-hand side are parts of the body's attempt to overcome the disease. And when we think about designing therapies, the treatments, the targets, we're going to target things on the left-hand side to block. We're going to try to improve the things on the right-hand side, create more of those responses to try to overcome the things on the left-hand side. It's been really critical to understand this difference between things that cause disease and things that compensate for disease. And there's a whole area of artificial intelligence that we're using called causal modeling to identify the causes and the effects that are going to allow us to make sense of these things and find the right targets for these diseases. It's also critical to understand that it's not enough to find a target, because despite our best efforts, a lot of the therapeutic ideas that we develop are going to fail. And we just have to recognize that from the outset and give ourselves a lot of shots on the goal, a lot of attempts to hit the same target. So when we think about this, we think about this not just about what we are going to do in the laboratory, but how we're going to interact with clinical trials. Because quite often, the vast majority, in fact, of clinical trials fail. And that's because we're just dealing with fundamentally really hard problems. And so when a drug goes to the clinic, you introduce into a small number of patients in different phases until you can eventually see whether it's having some therapeutic benefit. And when it fails, typically, there's been so much of investment in that one clinical trial that there's really no effort put in afterwards. Typically, when clinical trials fail, that's the end of it. But when we think about this from the perspective of engineering, we realize that there's a lot more that can be done here, because in engineering, we know that failure is part of our design process. We build prototypes, we test things out, and they don't always go the way we expect. But if we keep improving on those models, we can get to something that is actually quite productive. And in the engineering design process, we think about a cycle where we begin with some measurement, in our case, discovering what's wrong in the disease. And then we come up with a model, maybe it's a computational model, that helps us predict what to intervene on, how we might prevent the disease. And then we go back and we test. We put some change into cells, whether it's applying a drug or some genetic modification. And we test what the effect of that change is, that manipulation. So we go back and we measure. And the cycle of measuring, modeling, and manipulating is done many, many times until we can refine our interventions and come up with the right drugs. So we need to introduce this cyclic process into clinical trials to learn from what's going wrong in the clinical trial and maybe find some way to make better use of the drugs that are already in development. We think this can be done in clinical trials actually with all the technologies that I was just telling you about. So with the kinds of data that were gathered in Answer ALS, we can now begin to predict different types of patients based on clinical or molecular differences. And that allows us to predict ideas, to suggest ideas about what might be appropriate for different groups of patients. Now, we can't go directly into the patient, that's too risky, but we can actually go back into their cells, these iPSC cells, and use technologies that edit the genome or apply chemical screens and see how the cells respond to those different manipulations. Some of these ideas may be amazing successes, but most likely, most of these ideas will fail. That's okay, because we're going to go back and try to measure what did they do? What did they change in the direction we had hoped, and what did they not change? And cycle through this process many times until we can come up with the best therapeutics. This kind of approach is really fundamental to what we do in engineering of anything else, be it from water plants to cell phones. So why hasn't this been done already for dementia? Well, a lot of these technologies are really new and expensive. Induced pluripotent stem cells, genomics, epigenomics, proteomics, these are all very, very expensive technologies that have been developed very recently, as is the technology for interventions, kind of CRISPR screening, and certainly artificial intelligence to make sense of all this. The expertise for these technologies is often scattered, and so it's rare to be able to bring together experts from all of these different areas. We've been fortunate in our Answer ALS effort to be able to collaborate with some of the best minds around the world, best laboratories, to bring together both the clinical, the cell engineering, and the artificial intelligence technologies to really make a difference here. And so when we think about what's going to happen in the next few years, we really are quite ambitious in our goals. We think that the 20th century gave us the tools to conquer so many different kinds of diseases. And we can see right now playing out around the world the hopeful signs of turning the corner with something as terrible as COVID-19. And now we believe the time is for basic science to turn its attention to the next great challenge. We think that next great challenge is dementia. We hope that the 21st century brings us as big a change to dementia as we've seen for other really terrible diseases. Thank you so much for your attention. Goodbye.
Thank you, Professor Ernest Frankel. Although it is just a video presentation to the time difference between us and Professor Ernest, but here I try to make a note from his presentation, and it is about technology to cure many diseases, which is developed so fast these current years. He described how the disease developed from the past up to now, including infectious disease and up to the degenerative disease such as dementia, Alzheimer, ALS, and so forth, and also modern viral infectious disease. I said it is modern because it is developed in current years, such as HIV, and now we experience the COVID pandemic. He also described the development on how to cure cancer, and then in ALS and else. Then he also in the last part showing us the development of engineering and of techniques to cure the diseases based on the clinical data, and then how we should select what technologies is required to cure the disease, or if there is needed a combination of technologies, including gene editing, the current developing technologies in therapies. And also he showed us that the collaboration among scientists and health practitioners is also a necessity to cope many diseases develop in this stage. Yes, thank you, Ms. Begum. We are happy to find you here. And you drop the notes. How do they propose to use AI to cure dementia? That would be an interesting research. Yes, of course. And we will take care of your question, and we will extend to Professor Ernest. The committee will do, and then we will share the answer. to you. Dear participant, again, because Professor Agnes is not with us this moment, so would you please to write down your questions? We will record and send it to him. Well, we will continue to the second presentation by Professor Mario Guarancino from National Research Council of Italy. Professor Mario Rosario Guarancino, PhD. Actually, it is his second chance to give us a lecture in bioinformatics. I hope he's here. Is he here? The committee? No? He is a researcher at the High Performance Computing and Networking Institute in Italy. His doctoral degree was achieved from Department of Mathematics in Applied Mathematics from University of Naples, Federico, Italy, and his postdoctoral degrees is from National Research Councils. And his main research areas in bioinformatics, machine learning and data mining algorithms, methods and software tools for biological data analysis. From this scope, we can see that biology cannot be separated from mathematics and informatics technology. And his main publication, of course, he has already published not less than 100 papers. The main topics is in learning. It is, I think, this is the newest. Learning from metabolic networks, current trend and future direction for precision medicine. It is about personalization therapy approach. And then SIRMM, Searchable Information Repository of Mathematical Models. It's also very useful for studying biologies and biological fields, as well as the diseases we cope these days. So I think I will not talk too much, and his profile is already presented to us. So then we are welcoming the speech of Professor Mario Rosario Guarancino. Please.
Hi, my name is Mario Guaracino, and today we will talk about the potential of big data analytics for research. This is a very hot topic, and as we know, every day we produce an incredible amount of data. There are about 2.5 exabytes of bytes of data that are created every day. The speed is accelerating, and the growth of internet and the Internet of Things is even pushing further this process. When we talk about exabytes, we mean this type of figures here. So this is really a lot of data. And the fact that many objects are connected to the Internet, our phones, our devices, our cars, maybe our fridges, will even increase the amount of data that every day it is generated and delivered through the Internet. In the last 2 years, we generated 90% of all existing data. This means that this process is going on with an increasing speed. So the gradient of the data production is increasing more and more. And what we believe is that by 2022, there will be more bytes than 10 times the number of stars in the universe. To have an idea of what is happening, I like to look at this website, Data Never Sleeps. So, and it gives statistics on how much data is generated every minute. So what we know is that, as for July 2021, more than half of the people on our planet are reached by internet, and there has been a 10% increase from January 2021. Another fact that we know is that in 2021 there was 79 zettabytes of data that has been produced. Of course, the data that is consumed is much more, because a single object, a single dataset, can be consumed by users many times. Think about video. It is produced once, but it is consumed several times, sometimes millions of times. So, but what happens to this data? Who is really consuming and producing data? Let's look at these infographics. So this is the data that is actually produced and consumed every minute in a day. So every day, every single day of our life, there will be such a thing. And this is very interesting. For example, let's look at TikTok. Many of us, many of you, are users of TikTok, and every minute on TikTok there are 167 million videos that are viewed. And then if we look at Instagram, every minute there are 65,000 photos that are shared. And what about Facebook? 240,000 of photos that are uploaded and shared every minute. So, and YouTube, more than 500,000 hours of videos. So what we get from here is that there is a huge avalanche of data that is produced and consumed by people in every moment. Of course, this is not only in our life, real life, but also in science and technology. So what we would like to see today, what we would like to talk today, is how this is impacting on our research. So how it is really changing the way in which we do research. So, but let's give a definition of big data. This definition that I have here is the definition that you get from Wikipedia. So what does it say? It says big data is a field that treats ways to analyze, systematically extract information from, or otherwise deal with datasets that are too large or complex to be dealt by traditional data processing application software. So what does it mean? It means, from this definition, that if you have complex data, and we will define what are complex data, or you have data that are too large to be dealt with a laptop or a standard computer, then this is big data. This is for sure a part of the story, but in my opinion this is not completely representing the full story. So I have an alternative definition, and this is the one that I like. This is very simple, maybe simpler than this, but maybe looks at the problem from a different point of view. So what do we have here? We have that big data is when complex data completely characterize a problem. Independently of its size, they are big. So, for example, suppose that we have a whole genome sequencing experiment. Of course, this is plenty of data. This is in the order of gigabytes of data, right? And this is completely characterizing a problem. The problem, so the sequencing of the genome. So this is big data for one organism, but on the other hand, this is not big data for a population. So big data is big with respect to how actually it characterizes the situation. So how it characterizes a single situation. So think about a town and its census. So there will be in the town hall a place where we annotate everything that is, every people that has come to the town or has left, that was born or died. So this for the town is big data because it's completely characterizing its population. Maybe the town has only 5,000 people, Maybe there is another town that has millions of people. In both cases, the data from census are big data independently of its size. With respect to complexity, we should also say some words. This is due to the fact that nowadays the data that we are treating is very different from the ones that we have been used to treat so far, to analyze so far. So when I was when I was a PhD student, data was very few. So it was very difficult to get data. Usually we got an Excel file, a table in which there were some observations and some values. Nowadays, data is much more complex than this. First of all, it can be a text, for example, or it can be the content of a video, or it can be anything else, data that comes from a genomic experiment. Moreover, this data is very difficult to store in a very single place. If we think about the cameras that are, for example, in towns that are monitoring the traffic in many towns around the world, it is really impossible to store all this data. The data arrives in forms of a stream. This is also true, for example, if we think about sensors that have been installed on our cultivars. So in agriculture, very often we have sensors that are sensing the speed of the wind, the humidity, the insulation, and so forth. So this means that this data is huge and is also complex. We have many streams of data that are arriving, for example, at different moments, and it is for this reason it's complex. And we have to analyze it on the fly. It is very difficult. It is really impossible to store the data, to build our models, to make some characterization or predictions. So let me see if we can continue on this topic. So when we talk about, as we said, big data is characterized by its size. That is behind the ability of a computer, of a common computer, to manage and process this data in a tolerable elapsed time. So we have also to find new ways to integrate this data, because it is also true, as I was mentioning before with the example of data coming from sensors, this data might be very diverse, complex, and massive. So what we want to clarify is that there are some concepts that are intrinsic to big data. These are the volume, the variety, and the velocity. Actually, when we handle big data, it might be not simple to observe what is happening, because maybe the volume of the data is huge, or also the diversity, the variety of this data is high, or maybe it is produced at very high speed. So these are the three V's, what are called the three V's. But there are many more V's that are coming and that are actually characterizing what the type, the big data. For example, we should give great importance to the value of the data. Those days it's very common to forget the value of the data. And as we see many times in machine learning and in artificial intelligence, if a system is not providing the answer that we expect, if an artificial intelligence learns but the answers are not those that we are expecting, then what we do, we just pour more data. But maybe this data has some characteristics that make it very little informative. So it is also important to really understand the value of this. We have talked about velocity and volume and variability, but another very important topic is the veracity of data. So the question is, can we trust the data? And we have seen this so far with the spread of fake news on social networks, right? what we have seen many times is that sometimes data is not really trustable. So the trustworthiness of data is a very central topic in big data. So how can we trust this data? Who has produced this data? Are we sure that it hasn't been attacked by somebody in a malicious way so that then our models will be corrupted by this data? This is usually in the field of artificial intelligence and machine learning, it's called adversarial learning. And then also the variety. Variety is, we have been already talking about this. This is also a very, very important, topic. Nowadays, we analyze many situations, we analyze observation using some variables. And those variables are also interesting when we look at their relations, so if they are correlated, not correlated, and so forth. So having an idea of the features that characterize a phenomena, but also the interactions existing by those, among those features is very important. And those objects that describe variables and their interactions are usually called networks or graph. And this is a type of complex data. So we can, for example, describe a cancer patient using the metabolic network that is obtained from the analysis of his or her cancer cells. So the patient becomes a network, and we might have several networks that are part of our sample, and maybe we want to organize and analyze, characterize this data. And then there are probabilistic data, linked data, dynamic, and so forth. Also important are unstructured data. This is due to the fact that in those days, as I was mentioning before, data is maybe in written form, or maybe it is a video. If we think about the health record of a patient, the place where the medical doctor annotates the characteristics of the patients, the number, the drugs that he gives, and also what is his or her diagnosis, then we understand that every doctor will write this in a different way. And even the name of the disease might be slightly different, as there are several taxonomies that are currently used to organize the diseases to organize the diseases. So, but let's look also at how the data is growing. So on this left panel that I'm showing here, there are data and up to 2019 and the predictions that are obtained from this data for several fields of science. So, for example, the black line, talks about astronomy. So, how is growing, or how data is growing in the field of astronomy, sociology, and also the characteristic of top 500 supercomputers. This is a list that actually lists all the top 500 fastest supercomputers that have been installed around the globe, and also the genetics. So genomics is the red line, and as we can see, it has a trend that is increasing over time. And we believe that there will be this is the y-axis as a log scale in petabytes. So what we expect is an incredible increase of data over the years. If we look, but this is only one part of the story, because in the study of human beings, there are not only data that are obtained from sequencing experiments, but there are also data, for example, that are obtained from X-rays or fMRI, so biomedical imaging techniques. genomics. And also some other types of experiments like whole genome experiments and whole genome sequencing and whole exome sequencing and so forth. So as we can see, there has been an increase in this type of techniques and in their use, in their common use for both research and diagnosis. What about the omics dataset? So omics datasets are freely available, so we can download them and we can use them to try our techniques, our data analysis techniques. So if we look at the genomic, at the omics dataset, we see that they are constantly increasing over the years. Those have different characteristics. For example, they might be the result of a genomic experiment in which we are trying to determine or quantify the genomic aspects of an individual, for example, the gene abundance or the structural variation that is existing within a gene or some other characteristic that we have. And also we have the metabolomics data in which we quantify the abundance of small molecules within a cell, or transcriptomics. So, for example, we might be interested in identifying the isoforms of a certain gene, or its differential expression, for example, in two conditions. And also there are types of data that are produced by spectrometers in the field of proteomics. So, and if we look at the right side of this panel, what we see is that the number of datasets that are available in repositories is huge. For example, the datasets all around the globe, omics datasets, are estimated to be around 500,000. So this is a huge number, and they are divided, as you can see, among the various types of experiments that are possible. So what does it mean? It means that there is a huge abundance of data. And our problem is now to understand how to select those data. And of course, how we can integrate different data, because it is not very simple to take the data and to put it all together. In the sense, what I'm saying here is that taking together data coming from different experiments is not very simple. In the sense that the experimental conditions might be not well explained or different, and this will prevent us from putting together data, from integrating the data. Or even if we have been batching our data, so we have conducting our experiments, we have to take care of the fact that we are repeating the same experiments, but in different at different moments. So maybe the machine settings have slightly changed, maybe sometimes has passed and our samples are slightly different from the ones that we have been analyzing before, or the temperature, or the room condition have changed for some reason. So the integration of data, of datasets, is very challenging problem. In general, we have several omics datasets, and in this slide and in the following ones, I was collecting actually some of the most famous catalogs and databases that are available for genomics. Just to name one is the GWAS catalog and the international HapMap project, which aims at providing a catalog of human genetic variations on, or for example, the 1,000 and the 10,000 genome projects that have been sequencing thousands of healthy people coming from all over the world. to have a standard to compare with. So to have a standard a reference of healthy people, or at least of people for which there is that they don't know to have some disease, and that can represent the world population and how, which are the variations among people living in different parts of the globe. Then there are the transcriptomic datasets. For sure, the geo and the encode datasets are the largest databases that are available. And from there, we can download, obtained through chip sequencing or bisulfite sequencing experiments. So there are, of course, here again the data is freely available. Each of these datasets is in the order of gigabytes. So when we build our experiment, so if we want to test an hypothesis, then of course we have to think about how much data we can handle. So it is not true that we can handle every type of data and we can actually solve every problem. But to answer some questions, we have to decide which is the dimension of our dataset, the dataset that with our hardware and our algorithms we can analyze, and then we can start looking for the data and download it in the proper way. So the planning is always more important than the experimentation in itself. So planning the experiments should take a reasonable amount of data. These are some others. These are ensemble. This is a database of long non-coding molecules. This is LNCP that is also very, very famous. Those databases are coming out every day. So, and the important thing is to understand and to check when they were lastly updated. So, if they are maintained, if the curation is manual or if it is automatic, because many times data is poured in without any check, and this means that the bias that we will find, the errors that we will find, will be much higher than in the case that we have datasets that have been curated manually. So, of course, that is the story looked from the side, from the Western view of the story. But of course, also Asian countries are developing a very strong infrastructure for handling big data. As we have understood so far, who has the data and who is able to analyze the data will be capable to drive the innovation in all the world. So in this moment, all developed countries have initiatives that are devoted to understanding how they should develop their technological infrastructure in such a way that they can handle their data and they can also analyze this data. And with the pandemic, we have seen how important it was the answer from governmental institutions or world institutions to try to fight with the current pandemic. So the governments that have been trying to collect the data, to plan a collection of data and the distribution or a wide distribution of data, have had more chances to develop strategies that were more tight, more effective with respect to those countries in which this effort hasn't been strong enough. So again, having the data and being the owner of the data and being able to analyze it is, of course, very, very important. As I was mentioning, this is BIG, so the Beijing Institute for Genomics. This is one of the largest installation in Asia with respect to biotechnological research, and they also have a huge amount of data. I think they are one of the largest facilities for genome sequencing. So they are increasing the speed, the pace at which the genomes are sequenced, and then they also developed their own technologies to have actually to have this data people sequence. So as we can see here in this picture, there is the resources that are made available by the National Genomics, by the National Genomic Data Center. So as I was saying before, we were talking about velocity. So we said the velocity is something that we should take into account. So at the moment, the human genome sequencing still takes more than a day. So in general, to produce from the sample to the FASTA file, let's say the final file that you get with the sequencing inside, it takes much more than 1 day. But people are pushing so that we can sequence the genome in less than 24 hours. But imagine that you could have a genome instantly. So you could go to a person, maybe take a cell or a sample of his or her blood and get the genome. So what could we do with those data? And are our algorithms, our methodologies, our software able to analyze this data at the proper pace, at the proper speed? So this is a very challenging situation. So how would our understanding of human health, for example, but this is also applicable to, for example, cultivars or other situations in which, for example, we have animals for feeding people or whatever. So how would we change our understanding of phenomena if we were able to instantly analyze data that is instantly produced? So of course this requires there is no answer for this right now. So this will require an incredible amount of resources, both people working on this, but also hardware and software, networks, and all the technological infrastructure. And this, of course, might make us think of what will be the innovations that we will have in the near future. So we should start trying to think in a way in which we try to predict what is going to happen. So what if this technology is available now? This is not my invention. I remember that 20 years ago I was working with physicists, high energy physicists that work on very simple particles from our universe, so on very small particles. And 20 years ago, more than 20 years ago, they were thinking of what would have been happening today. So we saw how fast the speed is changing with respect to data generation, data consumption, and so forth. So how will we be able to analyze this data in the future? To make just a simple example, the other day, and just actually to nearly conclude what I was going to say, the other day I was looking at Instagram and I was looking at some photo of some friends. And there was one friend that had uploaded a photograph in which he was having breakfast. So there was cappuccino, there was like some cakes and so forth. And on the bottom of the picture, there was a banner saying, if you are looking for pandemic news, for pandemic data, please check this website. So it was a trusted website, and this was automatically added by Instagram to the picture. So at the first moment I was like, why are they talking about why Instagram is pointing me to this resource if this photograph is talking about a breakfast? But I looked at it again and I saw that there was also a newspaper. So in the photograph, my friend was having breakfast and was reading a newspaper, and one of the titles of the head news that was in the newspaper had the word COVID inside. So this means that Instagram is analyzing every single picture that is uploaded, and we have seen how many pictures per minute are uploaded, and is checking for for the content. In that case, the artificial intelligence was detecting the word copying, so it was providing a link to trustable resources. So, of course, this requires a technology. This requires also the fact that the data is yours, so you can analyze this data. So if we move this reasoning to biotechnological application, we can pretty well understand what is the situation. So I already explained how important it is to understand what are the computations that are possible as at a different velocity of generated data. But I wanted to close showing you a very last picture that talks about the variance, the variety of sequencing assays, because every type of data requires a different algorithm. So, for example, if we have a genome experiment, this will require a very different algorithms and software for the analysis. And if we have a proteome or transcriptome or whatever type of experiment, we have to adapt our methodologies. Just to conclude, I think that in this we are very lucky to live in this moment in which there are so many challenging problems. And we are in a moment in which we can more or less predict which will be the development of big data. So how the things will increase. So we have trends that we can analyze. So our purpose, our main objective will be to develop novel data analysis technique that can handle such complexity.
Thank you, Dr. Mario, Professor Mario. And again, I write down some notes from his presentation. He's presenting to us about big data, what is big data and how is big data developing, and how the data never sleep. It is kind of a software, I think, but the name is so intricate, and of course, the data never sleeps. In a second, we close our eyes. The data add in an abundant amount. And also, he explained to us the impact of the big data and how it has changed our behavior in research. We know now that we cannot do any research without searching every possible resources, telling the problems or idea we will do in the research. So then he also described what big data is. From whole genome sequencing, it is big data for one organism. It is the limitation, but not for all populations. So then we need to understand what is the definition, the true definition of big data. Also, he explained to us about the volume, variety, and velocity of big data, which is then required us to embrace the computational approach to understand our data and also supporting data we need in our research. And also, he also opened our eyes that there are many approaches to get the data. And then also, I learned still there is humanity in this speak about big data, which is then a scientist let us access the data freely. They shared it in some websites providing the data of any omics or even every omics we need, whether it is proteomics data or transcriptomics or genomic data, and also metabolomics data. Then we need to search, and also we need to learn ourselves, to develop ourselves, to be able to be in this era, the bioinformatics computational approach era. And he also depicted here how a scientist developed himself. It means that how we, from the wet lab based scientists, then we should embrace a computational approach, including bioinformatics approach, to understand our data and to analyze our data. There is a presentation I could wrote down from Professor Mario Guarancino, and last notice that he said we are lucky to live in this moment with a lot of problems in health and life, but then the data, the technologies is also provided for us to be used to understand this problem and the disease, and we then can develop our approach to cure the diseases and to solve the problems. It is the presentation from two professors, Professors Ernest Frankel and Professor Mario Guarancino. From them, we understand that, what technology is developing to cure the disease, and then one approach to understand the data by means of computational biology and bioinformatics. And then we will welcome Dr. Riza Putranto, which in daily life now always share with us his views and his knowledge in how bioinformatics help us to understand the current situation, the current problems, and also the current disease hit us strongly as a pandemic. Here we will welcome Dr. Riza Putranto, which will present about practical application of bioinformatics in current health problem solutions. But before we let Dr. Riza to present directly to us his view, let us introduce him. He's a molecular biologist with specific major in molecular genomic and bioinformation. Researchers at High Performance Computing and Networking Institute of the Italian. No, it is not. The first part is not right, I think. He got his bachelor degree from Gadjah Mada University in biology, master degree from Université des Sciences et Techniques du Montpellier. I'm not good in French, Dr. Putranto, and also his doctoral philosophy in the same institution, in the same universities. His research area is molecular biology and bioinformatics, with application in the molecular genomics, transcriptomics, as well as metagenomics analysis, using real-time PCR and sequence polymorphism by GBS. His experience as a chairman in the Indonesian Society of Bioinformatics and Biodiversity. I think I should join your society, Dr. Riza, because my background is not in this field. Actually, I'm in animal development biology, especially in animal development. But in this current situation, I should also accompany students who's interested in barcodes for systematics and taxonomic studies and also in COVID. And also some of us following you in your Instagram account. So I'm not strange with you, I think. I learned a lot from your post in Instagram. So then participant, we are welcoming Dr. Riza Putranto to present his topics this morning. Please.
Oke, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, ladies and gentlemen, dear friends. I see some of my friends actually they are waving at me in direct message in my Instagram. Thank you. First of all, I would like to express my regards to the Dean of Faculty of Mathematics and Natural Sciences, the management of also the Faculty of Mathematics and Natural Sciences, the Department of Biotechnology, State University of Malang. I'm very happy to be in front of you, even though that we have to do this again in virtual mode through Zoom. I would like to express also my gratitude, and also thank you, Dr. Dwi Listiorini, for presenting me and reading my curriculum vitae. So this morning I was feeling excited to do this presentation, and for this morning presentations, I would like to share with you my experience, and also I will give you later on some examples on how bioinformatics can help us in the real world application. So I will tell you first before the story of bioinformatics in general, and then we can go to the different level of bioinformatics utilizations in real world. So Dr. Dwi has already introduced me. Basically, I work in several institutions, but mainly I am a research scientist in the Indonesian Research Institute for Biotechnology and Bioindustry. We work with many species, especially plant species, but also bacteria and viruses. And that's why I am, or I was interested in studying the molecular genomics, because inside we have this bioinformatics that is going on. And at that time, I was in France during the 2007-2014 for 7 years. And there I learned a lot about how bioinformatics can help my major, the molecular biology, to improve better to the research. So the outline of my presentations, I separated only in two. So basically we will talk about where is bioinformatics today in the real world applications also. And these two outlines will highlight all that I'm going to present in front of you. So first of all, we would like to talk about bioinformatics today. I hope that all of you has already taken your breakfast, because the picture might induce you a little bit of adrenaline or maybe epinephrine to make you want to eat more. But this is an example. I took just one of this Indonesian dish, or what we call laksa mie rebus. It's like Indonesian cuisine mixed with noodles and with meatballs or fish, or maybe vegetables and some spices. Like you can see, there is a peanuts also, and it looks very nice. It's the combinations of many things, of many stuff, like noodles, meatballs, fish, vegetables, and spices. And why I brought you this picture, because I would like to talk to you. Bioinformatics is actually look-alike like the mie laksa. So this is the plate of bioinformatics. When you Google bioinformatics and you search, for example, the National Center for Biotechnology Informations and CBI in 2001, they define bioinformatics as the field of the science in which biology, computer sciences, and information technology merge into a single discipline. Meaning what? Meaning that the old ways, I would say the old ways, but with like the old ways of molecular biology, like we are pipetting DNA or RNA in the laboratory inside a liquid, or maybe when you do a PCR using tight polymerases, a mixture of reagents, and many things, cloning. It's not only that. Bioinformatics now enters the world of molecular biology and even biochemistry. And they merge together, just like as you can see in the plate. You learn about DNA, you learn about proteins, you learn about RNAs. Now we have this pandemic with SARS-CoV-2, with the RNA viruses, and we try to understand what happened with these viruses, and we try to monitor the development of the mutations. I will tell you this story about a few moments later. And this has been combined with your computer, the algorithm that maybe mathematicians made, or even statisticians, or even the computer science researchers, that has been combined into the plate of bioinformatics. So this is not a domain or a subject that you can learn by only trying to learn the algorithm itself, or only the DNA or RNA part itself. It should be merging together to put this plate of bioinformatics in use. And that's what we see with bioinformatics. And we have seen in the previous presentations with the omics, we know that bioinformatics is there. So allow me to note the quotation of Dr. Janet M. Thornton. She's the biochemistry and molecular biologist in the Department of University College in London. She's the expert of crystallography. You know that crystallography is one of the methodology that we use to find out the structure of protein. And she said, for a biologist, bioinformatics expertise is no longer an optional extra. It's no longer a tool, but it is a core skill. If we want to benefit fully from the genomic data that you have in NCBI, for example, or DDBG or EBA UK or others databases, you need to establish adequate skills on bioinformatics, even from since the, what we say, young ages, from the undergraduates level upwards into, if you are interested, to your PhD. And this is very essential, she said. And what we learned from bioinformatics along these past years, because I started to learn bioinformatics in 2005, well, basically when I was accepted as a scientist. And when I was a young scientist, I understand that bioinformatics needs three indispensable aspects. The first one, of course, the databases. If you don't have the databases, the data, the sequences, you will not be able to perform bioinformatics. So database storage is very, very important. And this is the part of where, when you are a bioinformatician, you might in general be separated in two groups, like a structural bioinformaticians and a functional bioinformaticians. The structural bioinformaticians learn how to develop the algorithm. You build your databases, how to develop your apps, software, and other platforms or even pipelines. Even though the functional bioinformatician learn how to study the sequence, how to use the applications, how to use the software to be able to get the result analyzed. So these three things: first, databases, and second, the masteries of tools by the functional bioinformaticians. You know how to do mass, you know how to do gene matcher, you know how to do a pattern find, on even how to do a MIM analysis. MIM is the analysis that we use to search the cis regulatory elements on the promoters of a gene, and to predict if there are or there's some of this cis element regulatory that can be used or can regulate your genes. So this mastery of tools is one aspect, and other things is the interpretation of results. For example, for bioinformaticians that are truly 100% computer science, for example, they might not be able to understand what changes in amino acid can result. But the biologists or chemists can understand that. So this is why the world of bioinformatics should be the combinations of many domain and many subjects. And in the 2020, we catalog in the world for more than 4,000 databases involving more than 7,000 publications and even more than 2,000 organizations throughout the world. It means that bioinformatics is now known in the world, and many people or many scientists are going through it, and they try to study it and try to perform bioinformatics in their research. But mainly when you see the databases from these thousands of databases, you have or your colleague give you a sequence, and then you try to validate and validate. I can give you an analogy. You can see on the left side, you have this famous Darth Vader from the Star Wars movie. Darth Vader looks like this with this helmet, with this lightsaber sword, and with this voice that is really recognizable, right? So if you imagine Darth Vader is a sequence, unknown sequence from unknown species, then you try to recreate these sequences, this Darth Vader. You find the right part on the helmet, for example, like this. And then you try to recreate the body. When you find the body is too short, the sequence is too short, right? And it's look more fat than the real Darth Vader, then you need to validate, revalidate, and revalidate. This is bioinformatics. So bioinformatics is trying to get approach to the reality as close as possible. We know that some of the sequences in the world that you might just input it in NCBI might not real in the real species. For example, because we try to recreate many sequences through the technology of real-time qPCR, for example, or sequencing that recreate different types of reads, contigs, and also scaffolds. So it will recreate what we call the pseudo sequence. And that's, we need to validate and validate and validate. And when we talk about bioinformatics today, we cannot separate bioinformatics from omics. As mentioned by the professor previously, that today's omics is everywhere, data is everywhere, and today's omics becomes the foundation of advanced bioinformatics. Why? When I learned bioinformatics for the first time in 2001, and I became a real scientist in 2005, when we try to implement bioinformatics, we do it in a low debit style. What is low debit style? Low debit style is when you learn and try to study one sequence at a time, create a design primer for PCR one at a time, and then you try to study gene one at a time. So it's low debit. The high debit one is what we see today. We see the NGS experiments is very large. You see many people sequence everything. You see data generations, you see data analysis. And that's why from this analysis, you need the processing. The bioinformatics pipeline comes in and fills the gap. And with bioinformatics, we can study what we see or what we have. The data is so many. And then using bioinformatics, we just can arrange pipelines, arrange algorithm for it, and then we can see what we want to see. Because of why, when you talk about today's omics is all of this part of the central dogma of molecular biology. From DNA, you see genomics. From RNA, you see transcriptomics or xenomics, or and then from protein, you see proteomics. And then you have metabolomics, and then you have phenotype, phenomics. With all of this, creating many data, and then when we find, when you try to analyze the data, you need to process it using bioinformatics. So this is what we see today, and this is what we must accept. And in the future, I think it's getting more and more important to work on the higher level of bioinformatics. Let me give you an illustration for this. We know that the technology of the sequencing is really famous since the dawn of the sequencing of human genome in 2001 here. I hope that you can see the table, the figure is quite small. 2001 here, we have the price for about 100 million US dollar. And throughout the time, we see the reductions of cost per genome. Meaning what? Meaning getting cheaper and cheaper. Nowadays, you can just sequence or do an RNA sequencing for one sample for 5 million. In Indonesia, it's quite expensive, 5 million, but in France or in Europe or in US, it's quite cheap. So you can just sequence every single organism that you want. When you have your PhD study in one approach of the organism, you can just sequence it, very easy. So when people try to sequence everything, and the technology now, you have this small USB-like technology that we use today for sequencing the RNA of SARS-CoV-2, the technology from Nanopore, the Minayan. And you have also the bigger part of this technology from Illumina, for example. You will generate many data, and as expected, by any estimation, by Illumina itself, by historical persons, and by Moore's law, we can see the rise of data, or the tsunami of data. We talk now, until 2025, we talk for about one or even more than zeta base pair. Zeta base pair is a 1,000 billion. It's so many, it's 10 power 21. It's so many zeros, and you cannot imagine how many data is inside. So the tsunami of data needs something that be able to identify information that we need to know. And that is when bioinformatics helps the tsunami of data to be revealed. And yes, bioinformatics is the inseparable analysis of the NGS research. As you can see, every NGS research, I took this one example of the picture from Wang et al. In 2009, natural ref genetics. You can see here we have this mRNA from any species. For example, you made the fragments and then you put it into the adaptators, and then you sequence it and put it on the exonic reads. We call it exonic reads because, you know, mRNA only code for proteins. So all of these reads, you combine it, or we call it the assembly, after the process of the sequencing. Or here, for example, after the process of the sequencing, we have what we call the post-sequencing data analysis. What is that? It means that bioinformatics goes from here. When you have these exonic reads, they need to be mapped, they need to be assembled, they need to be visualized, they need to be quantified, because the quantification, the visualizations, all is the important part of this next generation sequencing. Without bioinformatics, you cannot combine all of this. You can only sequence, and then you have this raw data that you cannot analyze, and it could be the end of the sequencing, because the sequencing itself is one part. Bioinformatics itself is one part. You cannot do sequencing without bioinformatics. You cannot do bioinformatics without the sequencing. So both of these skills are needed. Matter of fact, many people, many scientists, and many students or PhD students try to improve themselves by learning the wet lab part and also the dry lab part. They learn how to do sequencing. They learn also how to perform the bioinformatics analysis. And other than sequencing, we know that bioinformatics is known for more. Maybe you can imagine that the multiple sequence alignment is the most common one. You have one sequence, you would like to understand what is that sequence, and then you put it in the BLAST analysis and in CBI, and you get the result. Or even you have this one sequence of proteins, and then you want to understand the type, the structure of the protein, you perform a protein modeling, or even, for example, what we did these past 2 years is the comparative genomics on how we try to understand the variant detections of SARS-CoV-2, for example, try to perform the drug design antivirus for SARS-CoV-2, transcriptomic analysis to understand the differences between the patients that has the severe disease of COVID-19 and the patient that did not have the severe disease of COVID-19. And even though we have a lot of utilizations of bioinformatics in CRISPR-Cas9 synthesis, for example, metagenomics and nutrigenomics, that is what we know. What we haven't done in the most part on this world with bioinformatics is that artificial intelligence, how bioinformatics with automatization can perform artificial intelligence to understand, for example, which variant that will go dominant, for example. We haven't do that yet, and we are not yet have the capability to do it. Machine learning, concept of brain and high-level functioning, simulation of viral infections, that is the one that we would like to aim in the near future. So now a little bit of examples in real-world applications. And first of all, I would like to show you this image that bioinformatics is a powerful domain or subject to spot important information from mountains of data. So you see a mountain of data, you would like to know which one is important for the hypothesis that you are willing to answer. Bioinformatics is the tool. I would like to took an example of our own research collaborating with the Beijing Institute of Genomics and also CIRAD in France in cobra genes. So basically, in short term, briefly, we have these cobra genes in rubber tree. You know, rubber tree is one of these commodities in Indonesia in the plantations domains. It's very important because the natural rubber that produces by the tree are used in many industries, especially in tire industries, the planes, cars, right? Rubber bearing for your buildings, rubber bearing for your bridges is very important. So the demand in the world is very high. So every scientist tries to uphold the production of natural rubber, from its best clones to the less best clones. So basically, we sequence the genome of the Hevea brasiliensis or rubber tree, and we would like to know which part of this genome, which gene are involved in this regeneration of the stem. So you can see in the picture on the right side, these are the tapping panel. We call you with your knife. You can just swift upon the bark or the stem of this rubber tree. Then we'll have the flow of latex on white. The problem is that some of these plants can regenerate fast. Some of the clones for rubber tree doesn't. Meaning that there is a important aspect in genetics that can regulate them. So we try to take an example, took samples. That is a team from the Beijing, took samples from latex and also from bark here, bark, latex, and they try to sequence it. So the sequencing use mRNA, so it's transcriptomic data. They try to map, they have these reads, right? They have map reads, and they have this quite a good example of map transcriptomic against the genome of the rubber tree. So these are the data that you can find in NCBI. You can find it, just Google it, not Google it, just open NCBI, and then you can go to the SRA, the sequence read archive, and then you can just put this accession data. You can download the data, it's quite big. I think it's 7 giga per data. So you can download the data, and then you need to understand and find out which cobra genes, because cobra genes known in Arabidopsis to be involved in these differentiations of this stem, right? So you have this mountain of data, you combine with bioinformatics, and you try to find in between these few genes of cobra-likes, from cobra-likes A to cobra-likes P, which one that are involved in these mechanisms of the stem regenerations. So what we see here, these FPKM numbers, is the numbers of the expressions of these transcripts in the RNA sequencing. You can see here for the samples of bark and the sample of latex. For these two genes, HB, Hevea brasiliensis cobra-like E, and Hevea brasiliensis cobra-like P, you can see that it doesn't express in latex, so meaning that it's expressed only in bark. We have these two candidates, right? The p-value is significant, so we can trust the data because we use an algorithm to find the expression of both of these genes. And then you try to revalidate it using other samples. These are the samples that are treated with ethylene. So another point of rubber tree is that when you need to produce the latex, you need to induce the tree using a ethylene. Using ethyl, you can just put it in a liquid on the tapping panel, and then you have the flow of latex. So we try to to find it if there is a tree that is not treated, because zero, and treated with ethylene. You can see is that actually there is no value that is significant, only this HB cobra-like H that is nearly significant, because we put the p-value lower than 0.005. So meaning that first we find two genes that are regulated specifically in bark. So maybe these two candidates can be used as a marker such as breeding. And then we see another information is that the genes, all of the cobra genes from A to P, did not express in latex, meaning that it's specifically expressed in bark. So we find these candidates showing that from mountains of data, we find two genes that potentially we can continue the research to be used as a marker-assisted breeding. This is how bioinformatics spots which genes are related to traits what we want to see. The secondly is that bioinformatics is also powerful tools, not only to spot or highlight the one that we want to show, but to spot the differences from the mountains of data. And these are the way that we did since the beginning of the pandemic, how study of SARS-CoV-2 mutation was carried out. You can understand again, I show again the slides 3 times today, the central dogma of molecular biology, DNA, RNA, protein, phenotype. The virus is the RNA virus. When the changes in RNA happens, the virus can have the changes in protein related to its structure. First of all, the protein related to structure, structure related to phenotype. So phenotype is a form of organism. When you have the changes in this RNA, you have the changes of 3 aspects of virulence. First, media always talk about this, transmissibility, how fast is the transmissibility of the virus, how much people go into hospital, increased hospitalization, how much the possibility of the escape antibody and vaccines. Until today, we still don't have real data of this because we only have the modeling. But the 3 aspects of virulence, what we see from the mutations. People will ask, mutation is not always bad. Mutation can also be good because mutations can also have a bad influence to the viruses and viruses can just extinct. But the mutation sometimes in a small part and small portion, like we see, we only have 4 VOC, variant of concern, and only 2 VOI, variant of interest, but it can cause pandemic, right? Because it can adapt to the human host. So how the study of SARS-CoV-2 mutation was carried out? So it's using the DNA sequencing because this RNA, basically we do RNA sequencing in the virus. We use this technology, both, I just put 2 that use more often in Indonesia, Illumina Next 500, 250, and also the Minion Nano 4. And what we did is when we took the samples and we do sequencing, we do it what we call genomic sequencing. We generate the data like this using any apps from bioinformatics, and then we can differentiate from variant A, B, and C. If some of you familiar with GIS8, we have more than 4 million genomes in GIS8 per today, 29 October. everyone in the world, every laboratory that is able to do or perform sequencing, try to do this and try to understand and try to monitor the virus, because it's very important. Why? Because we need to make sure that the small portion of the viruses did not cause a huge problem in the world during the pandemic. And I will took one example of real-world applications in genomic surveillance did by the Koch UK consortium. The viral genome sequencing used to detect what we call the cryptic transmission, because you know SARS-CoV-2 with this in Bahasa we call it OTG, orang tanpa gejala, is asymptomatic patients is quite the transmissible one that can just go and waving around everywhere and just spread the disease. So the Koch UK consortium or the UK now have been massively sequenced these SARS-CoV-2 genomes to carry out case tracings, and they have come out with a number more than 1 million genomes in GIS8. It's more than 24% of the world SARS-CoV-2 genomes. How they do it? Basically, they try to do a case tracing. For example, finding the patient's X, right? The mutation of SARS-CoV-2 are known to 2 bases per month. So basically, when you have the changes on the genome, I took a small portion of the sequence like this. You have this virus A, patient 1, patient 2. When you sequence the viruses from these 2 patients, you did not show any changes. And then the patient 3 comes out to your hospitals and you sequence them also. You see double changes, GA and AA, meaning that there is a leap of mutations, right? It's supposed to be two letters or 2 nucleotides per month, but this is 4 nucleotides per month. So basically, you're missing 1 patient, what we call the patient's X, that having this mutation of GAUUCC. The first thing that the UK did is that they traced this in a whole lot of individuals. Secondly, they try to track the origin of the virus, origins of the disease, try to identify the cluster. For example, the patient's 1 is a Chinese, a Wuhanian. Patients 2 is an American, patients 2 American who just returned from traveling to Wuhan. And then you have patients 3 and 4 came from France, or is a French and also a Spanish, that doesn't have any history in visiting a country of origin of pandemic, but they have these mutations doubled. So means that there is a patient X who traveled from Wuhan, goes to Europe, right? The patient X. When they find the patient's X, they identify the cluster where the patient lives in the UK, and they identified it, and they did perform some partial lockdown, and they try to contain the disease as much as possible. So that is why, so this is a really good example of how bioinformatics from the small part of the sequence, and you work it in front of your computer, and now we can use it in the real world applications, and very useful to try to stop the spread of the disease. Other things is that bioinformatics can help you finding the differences between the sequences of the SARS-CoV-2 to identify which variant goes first, which variants is the ancient variants, and which variants goes down into the next further adaptation, for example, you might say. The first thing that you see here is you have a Wuhan variant, and then approximately in the month of April 2020, we have a lot of media saying there is a D614G mutations. Actually, it's the variant of B1. Now the variant of B1 are inside every variant that we can find in the world. Mention it, Delta, you have mutation of D614, Beta also, Gamma also, Kappa also, and others. Because of why? Because of these variants are actually derived from the previous variant. How? By replicating inside the host and having these mutations throughout the time. And until now, the mutation is quite complex, and we can see changes in the variant. And the last time we see Delta as the variant that gave us any problem. So in other things, from the perspective of science, you can just identify which variant goes first, right? In the history of the family tree of the SARS-CoV-2. But in terms of cases, you can work bioinformaticians and also molecular genomists and also epidemiologists can work together to follow the rise of the cases and the down of the cases. As you can see here, these are the daily COVID-19 cases in Indonesia. When you see the July cases to August, we have this huge peak, what we call the second high wave in Indonesia. When you see the genomic aspect of it, you find the SARS-CoV-2 here. The variant, what we call variant Delta AY23, that rise up during the cases and went down during the cases went down as well. So this is how bioinformatics can help you. You just identify the variant. Someone sends you samples from laboratory. But Riza, I send you samples of sequence. Please identify which variant is it. Using bioinformatics, we can identify by comparing the sequence that we just received with the sequence already referenced in the China, for example, the Wuhan sequences, and we know which variant is it. And these are the another example of the use of bioinformatics in real-world application. The other thing is that bioinformatics is also a powerful domain to visualize and model potential target drugs. For example, if you see this sequence, these are the sequence of SARS-CoV-2 main proteases. You see this protein, right? Methionine, arginine, lysine, etc., until the codon stop here. Bioinformatics can help you to convert this into this, the visual aspect. You can see the protein itself will look like a shape of a heart. You see the red part of it is the active site of these proteins. Then when you wanted to have these inhibitors to target the SARS-CoV-2 main proteases to make the virus cannot replicate, so you must target the red part. So they did here with the many possibilities of docking combinations. And when you find out this, I took an example of this only one study, but actually you can find more than 3,000 studies carried out from 2020 to 2021 in the PubMed.gov. So many studies about how main proteases needed to be blocked is already going on, and we hope that we find more and more potential drugs to be the antivirus of SARS-CoV-2, helping the vaccine. And the next thing is that bioinformatics is a powerful domain to ensure the successful genetic engineering. We went away from SARS-CoV-2 now, and I would like to show you how bioinformatics is indispensable in CRISPR research. It's going to be it's the current research that everybody wants to do. The CRISPR one. Took an example in cacao tree from our colleague Pfister and friends. He tried to perform or enhance the immunity of cacao against fungi, right? That is the basic knowledge. How? By targeting one gene, what we call the TC theobromocacao NPR3. NPR3 is the genes that are involved or in the defense mechanisms against the attack of fungi. Bioinformatics is there. How? Because we cannot perform any CRISPR research without doing first the modeling and also the design of this single guide RNA that will cut the DNA using bioinformatics, or even we cannot design the plasmid of CRISPR without using bioinformatics. Bioinformatics design these plasmids of containing Cas9 and also the single guide RNA here. They use two single guide RNA to ensure the KO of the cacao genome of the TCNPR3. They target the genome in exon 2 and exon 3 to ensure that the protein cannot be produced, and because NPR3 is the negative regulator of many immune systems in this cacao. When they get knocked out, the immune system rises up. It's like you open the channel of a water, and then the water can run through. So the immune system of the cacao getting improved, and they now are testing it in the field to see if the edit plants are also immune to the fungi disease. So if we can get back to the plate of bioinformatics and to end my presentations, actually today's bioinformatics that I show you do not play on small scale anymore. It plays on data integrations, or what we call multi-omics central hub. And the several examples of real-world applications in bioinformatics show that it can serve inside mountains of data to find the one that you want to highlight, to find the differences between the sequence and the data, and to identify and to use these differences on the applications that are really, really important in the daily life of a human. And during the era of pandemic, bioinformatics has shown its importance, so equals to other domains such as molecular biology, and even inseparable, because molecular biology without bioinformatics, you cannot do a what we call high debit, and bioinformatics without molecular biology, you cannot have the sequence and you don't have data to analyze. So to end my presentation, I like to acknowledge all my fellow scientists in IRIP, also young friends in Alibi, friends in Mutasi that we discuss every day about the mutations of SARS-CoV-2, Professor Chorong Tang from Beijing Institute of Genomic Catastrophe from allowing us to use the genome of Hevea brasiliensis, friends in crowdfight COVID-19 since March in 2020 until now. We still have discussions and performing analysis of SARS-CoV-2, MABI, Indonesia Genomics Consortium, BRIN, Ministry of Health, of course, the ones that has the data, GIS8, NextStrain, Regeneron Pharmaceutical, and also COQ-UK Consortium. So I'm open to discussions, and thank you very much for the opportunity. I go back to Dr. Dwi Listiorini. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Riza. Such a very interesting and also intriguing presentation, since a lot of the information you presented are still very new for us, I think, and also for me. And from your presentation, I took some notes that now we cannot live without bioinformatics, something like that. And bioinformatics helps us to understand the nature of organism, the phenotype, the metabolism, characters, and many things, and also the characters of virus, which now becomes a lot emerging. I think it will be more in the future. Don't you think so, Doctor? And also with the help of bioinformatics, we can alternate and improve the quality of organism, whether it is their products or their health, and also, of course, for ourselves as well. And with bioinformatics, we can a lot we can do, including studying, analyzing, designing, and also confirming the interaction between molecules. So in the past, I chose biology because I'm afraid of mathematics and physics, but now I cannot go away from this, and this a lot of interesting things. Then here we find a question from Bapak Putut Rahmat. Do you want to read or yourself, or I read for you?
Pak Putut Rahmat.
Pak Putut Rahmat in question. Both bioinformatics and I will read for you. It is okay.
Oh no, well, I can read it myself. It's okay. Okay.
Yes.
Yeah, because I see in the chat box. Actually, it's in the Q&A box. Okay. Thank you, Bu Dwi Listiorini. So the questions is from Pak Putu Rahmat. Okay, bioinformatics and computational biology are commonly known. That may confuse. So the first question, the differences between bioinformatics and computational biology. Bioinformatics is limited to sequences, if you would like to simplify the terminology. The macromolecules, DNA, RNA, and protein for now. That is what bioinformatics in. Computational biology is different. Computational biology, for example, if that you, I can take an example in ecology. You go up in the tree for days. I did it once when I was a student, and you make a video of the movement of the wild boar in Borneo at that time. You stay in your 3 days using cameras, still cameras that you put on your tree, and then you will see the movement of the animals. And then you make a model from that. You make a model how the animal will move, how the animals move in groups, where is the kids, where is the small boars, for example, and then try to make the circulations on how wild boar try to find the food, for example. That is computational biology, because you don't use sequences. But if you use sequences, for example, DNA, you would like to identify what is this DNA. It is a gene, it is a untranslated regions of an organism, then you do bioinformatics. I think that is the simplest explanations that differentiate between bioinformatics and computational biology. And the second question is that, okay, which one is first? Molecular biology first, or informatics first? For me, in my case, I learned molecular biology first. And bioinformatics goes second. But I think there is no regulation that you have to learn informatics first or biology first or vice versa. You need to learn both. Even you need to learn another type of science also, what we call visual science. Now in the era of digital, you need to have that also. You might have the capability doing in laboratory, such as you prepare yourself the samples for sequencing. That is the molecular biology part. And then you need to have the capability to analyze the sequence that you resulted from your own experiments in laboratory. You analyzed it, that is the informatics part or informatics part. And you need to be able to visualize it, chart, like a simple chart or tables that easy to people to understand that. So I think bioinformatics is now shifting also to visual sciences, because everything about bioinformatics is good and see it. If you show people or show students your data, your raw data with the numbers and algorithm using Linux, for example, they will get confused. So that is why we need to combine all of these possibilities. If not, to learn every only just of the do bioinformatics part, but not the wet lab part. It's okay. We can collaborate. That is the important things, I think, in this era, to collaborate also with other scientists. I hope that I answer Pak Putut. But if you have more remarks, I would glad to hear them. Thank you.
Thank you, Dr. Risa. So from your answer, it is depend on each from where to start the bioinformatics, right? And I think it is starting from ourselves. For example, we in UM starting from developmental biology and then from biostatistic. Then we enter the barcodes when the DNA barcodes coming to Indonesia by 2010. I say like this, I say for this. And then they're developing into other sides of bioinformatics. I can see that bioinformatics has many phases, something like that, Dr. Lisa. There is barcodes, there is docking, there is protein modeling, there is analyzing, and so forth, right? So for Pak Putut, it started from your field, from your specific field, doing your study with a bioinformatic approach. This is what Dr. Putranto explained. And then we have the second question from Nicholas Gary in chat box, please.
Okay. Thank you, Putri. I will read them. I have already read the question from Nicholas. Actually, he basically is asking about the disadvantages of bioinformatics. What are the disadvantages of bioinformatics? So I don't know if you know this, but we have in bioinformatics the term of GIGO, the garbage in, garbage out. What it means by that? What it means by GIGO, it means that if you have the sequence that you analyze using bioinformatics, but the sequence actually is a garbage. Why is it garbage? It resulted from fragmented DNA, for example, resulted from a poor quality of sequencing. Then you have this sequence that is not in a good quality, what we call a garbage. And the garbage is in because the bioinformatician and bioinformatics domain do the analysis part. And then it come out with the conclusion that these are variant X really dangerous, for example. But the garbage already in, so the one that is out cannot be reliable. So that is I think the disadvantages of bioinformatics, the disadvantages that we need to overcome, that we need to prevent. That is why it is very, very important to make sure before the bioinformatics is a sequence the quality validated. For example, you can see many reads. For example, if you have the sequencing, we have the quality check, the depth of sequencing, also the length of the sequencing. If you have many reads in one part of contig, you can believe that the sequence is real. But if you only have two reads there, you have this contig here, you can imagine that perhaps it could be derived from mistakes from sequencing. So the quality of the bioinformatics relies on the first part. If the sequence is not good, so the result is not good also. And what are the highlighted breakthroughs and great findings from bioinformatics this far? I think the pandemic is GIS8, you can see it, NextTrain, the covariant.org, Regeneron, Outbreak.info, and others, many things. I think that is the breakthrough. I never seen in my career as a bioinformatician someone or some team get databases as huge as now. You can imagine GIS8 has 4 million sequences already inside their servers. And the beginning of the pandemic, they only have several thousands. And throughout this pandemic, we multiply the amounts of the sequence. And I think the approach should be done in other diseases, for example, HIV or others. We need to monitor the mutations of the virus or bacteria even. And this can be a good example on how bioinformatics really can speed up the research of molecular biology. And the last question is that if you know, how does one develop an AI software to process biodata automatically? In Indonesia, I never talk about people doing this AI software process biodata automatically, but in Italy, in the St. Anna laboratory, I saw some some research scientists try to perform this, but I don't know if they have already published the paper, but maybe I will find out later. So thank you for the question, Nicholas. I hope I answer your your questions. Thank you, Budi.
Yes, you may take a little drink
Yeah.
just to fresh. Then the next question is from Dwita Oktiarni in question and answer box. Is
Yeah.
it possible the third wave will coming soon for this pandemic? Oh, it is about COVID.
Yeah, it's about COVID. It's the question of many people asking. I understand that we are now in the loosen era. I will say loosen time. So we feel relaxed, right? Everything, we even I have to always the term of the third wave in the pandemic for me personally is that since the virus cannot eradicate it from this world, so the possibility of the third wave is still there. How third wave can come, it depends on how the mobilization of people, mobilization of the populations. If we can limit or we can regulate the mobilization of the people, we can minimize the potential rise of the third wave. Because now we see that the Delta variant itself has already evolving a little bit, becoming more than 24 and 26 subvariants. What you see is with the number of AY, Y1, 2, 3, and Indonesia have 4. In UK, you have AY4 that is now becoming the headlines of every media. So every time you have these new variants, if these new variants is adapted well with the host in the specific locations, for example, in Indonesia, you might have this third wave. But to answer certainly these questions, we cannot, because it depends on how the mobilizations of the population is going, how the new variants can enter Indonesia, for example, or how if this variant in Indonesia, what AY23 developed into others or mutated into others variant, we don't know, because as I mentioned in my slides, there is one technology that we need to overcome is how to predict mutations of viruses, not only viruses and bacteria, in which exact moment that is the questions of all research scientists are looking, because we don't know when, right? Suddenly in July, we have this huge second wave, and then in August, September, you see the downfall of the cases. And even when we talk about friends in Next Strain, COG UK, seeing them talking in seminars, they even don't know how or in the future how to predict the mutations of virus. It is the questions of the current technologies that are limiting us, but in the future, maybe we can have this. So I hope I answered the question of Dwi Oktariani about the third wave. So it depends on our mobilizations, regulations, and the new variant. If all of these three aspects are met, then you will have the third wave. But I hope not, because it's going to be quite difficult for all of us. Thank you very much.
And there's still 3 questions for Dr. Riza, but I'd like to ask the committee, is it possible to continue, or how? Dr. Putranto still have a high spirit for us. We continue.
Okay.
Because there is a question. I try to
okay, maybe
stay fair. There is a question from Yudrik Latif in a chat box. What is the program can make a sequence of 3D view? I follow the sequence of the question who's coming first, answered first.
Okay. Okay. Thank you, Budwi. This is the question from Yudrik Latif. What is the program that can make a sequence to 3D view? Yeah. You have many. I use Fire2. The word is PHYRE2. So Fire2. maybe I can write on the chat box to everyone. Fire
Fire
2.
Oh,
Yeah.
it's not Fire
You
X.
have this also Swiss model. Yeah,
I
Fire
see.
X, you can use it also. You have, I forget the name of the other one, but you have to, but both, two of these I use it very often. Basically, I like the algorithm of Fire 2. You have blind sequences of protein, of amino acids, just put it in Fire 2, runs it, you have the models, and then you take the models out, download them, and then you open the model in Fimo. Use 3 of them, you can make a good paper. Because in Fimo you can make animations, you can make a different coloring like the one I showed you in the slides. You can show the active site of it, and Fire 2 using analysis can identify you pockets. Pockets is the active site of the protein, can identify you the bad shape of the protein because it's model, right? The one thing that you need to understand, Pak Yudri Kreatif, is when you do a modeling of a protein, you need to understand that some of these models might be wrong because the shape of amino acid, because amino acid is the 3D molecules. So when you put them together, it's like when you putting a bridge of cards. When you see one card is not meant to be there, it's going to be crumbled down. So when the modeling is not to find the perfect shape of the proteins, but to find which one is correct. So sometimes using Fire 2, you can have these colors differences in the analysis, green and also red. Red means it's bad, green means it's good. But now I think the color changed. Red is good, blue is bad. So you can see the one that you think is bad, and the one the one that Fire 2 thinks is bad, and the one that Fire 2 thinks is good. And can we make an functional analysis of it? Yes, you can do molecular dynamics after it using VINA or VMD mole dynamics. But this is a next step of bioinformatics. So when you do bioinformatics, when you're a beginner in bioinformatician, you need to start with multiple sequence alignment, pairwise alignment, creating design primers, phylogenetic analysis. That is for the beginning when you learn bioinformatics. And then you move forward to the comparative genomics. And then you move forward to the molecular docking, molecular modeling, protein modeling, and then molecular dynamics. I did in some, I think I posted one of my result in molecular dynamics in my Instagram. It's the picture of spike protein of SARS-CoV-2 getting close to the receptor of, I forgot the name of the receptor. the ACE2, sorry. The ACE2 receptor in human that receive the spike protein from SARS-CoV-2. That is molecular dynamic. You put it in the, for example, in the environment, you create an environment, but doing a model, for example, 33 degree, 35 degrees Celsius, and then you can see the molecules moving. That is molecular dynamics, and that is functional analysis of what your protein can do. I hope that I answered the questions of Pak Yudri Kreatif. Thank you. Bu Dwi, should I go on to the questions of maybe Sarina Bikam?
Sorry, Dr. Riza. I think Budwi has a bad connection.
Okay.
Yes, you may go on answering the question. There is some question left.
Yeah, there is I think 2 questions from 2 persons. So thank you, Pak Norman. I will continue to short the time. So Sariva Begum asks, is bioinformatics or more as accurate as conducting lab works using various biotests? in some cases, yes, in some cases, doesn't. In what cases? In the cases that you need to performing a huge analysis of a sequence like in pandemic, yes, it is accurate as the same one as the sequencing does. But for some reasons, bioinformatics cannot go past beyond its limit. For example, when you design CRISPR research, like the plasmid of CRISPR-Cas9 and also the single guide RNA in one plasmid, you can design, you need the last, and it's the limitation, I think, of for some bioinformatics are the helper of molecular biology, and for some molecular biology are the helpers for molecular bioinformatics. I hope the first questions, and the second question, all the disadvantages of bioinformatics in evaluating and predicting COVID-19 data for future predictions. So for now, bioinformatics did not use to predict mutations, but it used to identify mutations of the virus. Bioinformatics hasn't done anything yet about the predictions of the future pandemics, but epidemiologic, yes, because they use algorithm and also modeling to perform that. But bioinformatics stays on the sequence, stays on the mutations. But if they can do it, what are the disadvantages that you need to validate and revalidate? That is always the disadvantages of bioinformatics, because you need to revalidate using different algorithm, using different apps, different softwares. So when I don't know if you know, but we have this huge platform in the world, what we call Galaxy bioinformatics platform. You can Google it. We have servers in US, in Europe, in China, in Philippines, even Indonesia. We have servers for Galaxy, and inside this Galaxy, we have several or many softwares that you can use to validate your sequences. So when you make a simple explanation of this, when you try to make a phylogenetic tree, some students might ask me which type of tree building method that I should use. Is it the maximum likelihood? Is it the UVGMA, or is it the other things? So you can use everything. You make a tree from every single one, and you try to understand and you try to see the results. What we would like to see in the tree is that the groups are always the same, but the individuals or the taxa can be switched up. So I think that is the way that we to be done when using a validate and validate. I hope I answered your questions, Sariva, and I might go to the next questions from Febi Ermina. Okay, she asks, can we do a validation of CRISPR and the candidate DNA target with molecular docking? Yes, you may, but I have many scientists do this, but maybe because this sgRNA can be validated very easily using PCR in the wet lab. So maybe some of the people, because using docking, you add more analysis, that is bioinformatics. You need to go directly to the wet lab after you design things. So that is why this docking aspect of this CRISPR using the target, not many people do it. But if you would like to do a docking, you can find many apps. You can use MolDoc, you can use INA, even VMD, you do docking. You can go to Galaxy. I think they have many apps that can perform docking for you. And the second questions, I think that the questions are the same. The one in Bahasa, the other one in English. I hope I answered the questions of Febi Ermina. Thank you, Pak Norman.
Yes, I think that's all the question for you. And if there's any more question, I would like to say thank you for your eye-opening presentation, at least for me, because I was never able to grasp the idea on how to utilize the maximum potential on bioinformatics. But after listening to the presentation from Professor um, Frankel, Professor Guarancino, and from you, it's helping me a little bit more to understand what is bioinformatics and how to use it. And as our appreciation and our gratitude, we like to give you the certificate to the operator. Please assist me to share screen the certificate for Dr. Riza Putranto. This one is for Professor Ernest Frankel, and the next one we have certificate for Dr. Mario Guarancino, and the last one is for Dr. Riza Putranto. Thank you very much, Dr. Riza.
Thank you, Pak Norman.
Yes. And thank you also to Mrs. Dwi Listiorini, who unfortunately already left us due to a bad connection. So I will take care from this. So finally, Excellencies, distinguished guest participants, ladies and gentlemen, we are now at the end of today's event. Before I close this event, I would like to inform you that the certificate will be sent to you via email in a couple days, and I would like to remind you to don't forget to fill out the attendance link in the Zoom chat box. I think that's all from me. I genuinely thank you for your enthusiasm and your participation for today. I hope that we can learn so much from this event, and I do hope the very best day for you all going forwards. Please stay healthy. Good afternoon. Goodbye. And Assalamualaikum warahmatullahi wabarakatuh.
Universitas Negeri Malang offers the best education, research and community service in Indonesia. UM has consistently been maintaining its position as one of the best universities in the country. In 2018, UM is in the top 15 universities in Indonesia according to the Ministry of Research and Higher Education and 4ICU, which proves its quality as a center of excellence and reference. UM has dedicated itself as the learning university since 2009. It has strengthened its vision as a learning organization and as an outstanding learning resource in Indonesia. UM students are distributed to various high-quality and well-reputed study programs, extending from associate degree to doctoral degree programs. Among UM students, there are international students who come from various countries across the globe. Recently, UM has 45 study programs accredited B and 59 study programs accredited A nationally. UM was established on 18 October 1954 as a College of Teacher Training and Education, PTPG. The institution transformed into UM on the 4th of August 1999. UM's main campus is in Malang, surrounded by education facilities, recreation and tourism spots, sports and business centers. UM's Campus 2 is located not far from the East Gate of Malang. UM's Campus 3 is located in the Gate of Heritage area of the Shrine of Sukarno, the first President of Indonesia, in Blitar. Faculty of Education, Faculty of Letters, Faculty of Mathematics and Natural Sciences, Faculty of Economics, Faculty of Engineering, Faculty of Sports Sciences, Faculty of Social Sciences, Faculty of Psychology Education. Postgraduate programs. UM also offers associate degree in vocational education. They provide trainings in various skills. These are managed under the skill training programs for automotive, business, industry, and information technology, or known as PROBIS. Under the guidance of lecturers and experts in their respective fields, students can experience revolutionary learning innovations, develop their critical thinking skills, produce innovations, and nurture their enthusiasm for knowledge. These endeavors perfectly fit with UM's aspiration in becoming the best center for learning innovations in Indonesia. Universitas Negeri Malang is always committed to making a better Indonesia. At UM, we learn, do research, serve the community, and contribute to the betterment of Indonesia.
UM, the Learning University.